We are going to discuss part A, which is multiple choice question on chapter 2, which is gene technology. Which of the following tools in recombinant DNA technology is incorrectly paired with its function? So, which of the four statement is not true? Okay, so we are going to look at one by one, which is for A, plasmid. It carries foreign DNA fragment into a host cell. So plasmid is the most commonly used as a vector, okay, which is foreign DNA will be inserted into the plasmid, which function as vector, and then it will become recombinant DNA, okay, and then inserted into a host cell, usually a bacterial cell, okay. So that is plasmid. It functions as vector to carry foreign DNA that contains gene of interest. Okay, so statement A is correct. Okay, so we are going to proceed straight to statement C first. Okay, for the second statement, which is foreign DNA fragment, okay, that contains gene of interest. Foreign DNA fragments are fragments of DNA that contains information, or it is actually genes from another organism. It has the information to code and synthesize certain proteins that you are interested in. Okay, so it contains gene that you are interested in, genes that can produce certain protein that can become or code for protein that will become, for example, enzyme, okay, or hormones. Okay, so these are foreign DNA that you get from another organism, okay, of another species. And then statement D, host cell accept and allow the reproduction of cloning vectors. So earlier we looked at, okay, vectors are plasmid, okay, plasmid that initially we isolate the plasmid from a bacterial cell. Okay, so after you have inserted the foreign DNA into the plasmid, it becomes recombinant DNA, and then this recombinant DNA or recombinant plasmid you have to insert into a host cell. Okay, so usually the host cell are bacterial cell. So bacterial cell are capable of dividing, okay, into many copies. Okay, it divides to form clones. Okay, so as the bacterial cell divide, it produces clones of the gene of interest that you are interested in. And as the bacterial cell divides and produces many colonies of the bacterial cell, the bacterial cell is also capable of producing the product, which is the proteins. So statement D is correct. So for statement B, restriction enzymes. So restriction enzyme, it says here, joins DNA fragment with vectors. Okay, so as you know, the enzyme that joins DNA fragment is actually DNA ligase. Okay, so we have looked at DNA ligase during chapter 1, which is DNA replication. So the function of DNA ligase is to join, for example, Okazaki fragments into a continuous lagging strand, okay, during DNA replication. That is DNA ligase. So DNA ligase, we also used this enzyme in producing recombinant plasmid. Okay, so when you insert foreign DNA into plasmid, for example, here this is a plasmid, and then you insert a foreign DNA, the blue fragment here, into the plasmid. So you need to join these two different sources of DNA. So in order to join these two different sources of DNA, you need to join with the enzyme DNA ligase. Okay, so obviously statement B is not correct. And as for restriction enzyme, okay, a restriction enzyme is also known as restriction endonucleases. So these are enzymes that are naturally found in bacteria. So common example is EcoR1. So EcoR1 is the restriction enzyme that is found in the bacteria E. coli. Okay, so for all the restriction enzymes or restriction endonucleases, it will cut the DNA, either the DNA of the foreign organism that you are interested in, okay, DNA of another organism, or the plasmid. Okay, so the restriction endonucleases will cut at restriction site of the DNA, the plasmid or the foreign DNA, so it will cut at the restriction site. Okay, so every restriction enzyme has its own specific restriction site. For example, for E. coli, the restriction site that the enzyme will cut is it will read as GAA-TTC. So this is the restriction site for EcoR1. Okay, so it will cut the DNA uh, between the guanine and also the adenine. Okay, same goes here. It will cut between the adenine and guanine. Okay, it will cut in between here, and this will produce sticky N. Okay, so EcoR1 will cut the DNA in a staggered manner and produces sticky N. Another example here, you have the enzyme SMA1, okay, which is extracted from another bacteria. So this is the restriction site that the SMA1 will cut, okay, that reads CCCGGG. Okay, so it will cut straight and produces blunt end. So these are the two different types of ends that different restriction enzyme will cut. So statement B is incorrect. So that is the answer. A small double-stranded circular DNA found in bacterial cell is called as plasmid. Okay, so for example, within a bacteria, the bacteria will have a region, we call it as the nucleoid region. So this is where the bacterial chromosome will be located. And then the bacteria will also have another form of DNA, which is plasmid. Okay, so this is the plasmid. So plasmid is a small double-stranded circular DNA, and it will replicate separately from the bacterial chromosome. Okay, so plasmid is commonly used in the recombinant DNA technology. So plasmid is one of the tools used in recombinant DNA technology. So there are actually 6. Okay, so the first one is the target DNA. Okay, so the target DNA will contain the gene of interest, and then the restriction enzyme, which will cut the plasmid and also the foreign DNA at the restriction site. And then the third one, you have the DNA ligase. So the DNA ligase will join the plasmid and also the foreign DNA to form recombinant plasmid. And then you have and then the cloning vector. Okay, so the fourth one is the cloning vector. So the one of the most commonly used cloning vector is the plasmid. Okay, so plasmid are found in bacterial cells, so they are isolated from the bacterial cell to form the recombinant plasmid first, and then inserted back into the bacterial cell to allow the bacteria uh, to replicate. Okay, and then so the cloning vector, the function of it is to carry foreign DNA into host cell. So number 5, the fifth tool used in recombinant DNA technology is the host cell. So the most commonly used host cell is the bacterial cell. And then you need the marker genes. Okay, so marker genes are found in the plasmid. So for example, here, as you can see what I've drawn, here you have the plasmid. So plasmid will have 2 genes, okay, in the plasmid. So one of the gene is, we call it as selectable marker. So one of the example of the selectable marker are ampicillin resistant gene, which are found in the plasmid of E. coli. Okay, so the function of this ampicillin resistant gene is to allow the bacteria to survive in the condition where when you grow the bacteria in a medium that contains antibiotic, it allows the bacteria to be able to survive. Okay, so that is a selectable marker, example ampicillin resistant gene. And then another example, another region, we call it as the reporter gene. Okay, so one of the example of reporter gene that are found in plasmid of E. coli, for example, is like that gene. Okay, so what we have looked at during chapter 1, which is in the subtopic gene expression, where we look at like operon, as you can remember. So the like operon contains the gene like that gene. Okay, so the function of like that gene is to produce the enzyme beta galactosidase. Okay, so when the bacteria has the plasmid, okay, the in the presence of lactose, okay, the like that gene will produce beta galactosidase to hydrolyze or to metabolize the lactose into galactose and glucose. And within the region of this reporter gene, which is for example like that gene here, within the reporter gene you have the restriction site. Okay, so this is the site where the restriction enzyme will cut open the plasmid and insert the foreign DNA. Okay, so when you cut open the plasmid at the like that gene or the reporter gene, now you will produce the recombinant plasmid, and the like that gene now will not be able to produce the enzyme beta galactosidase. A molecule of cDNA is synthesized from, the answer would be D, mRNA. So what is cDNA? cDNA is complementary DNA. So C stands for complementary. Complementary DNA is basically DNA molecule made in vitro, which is in the lab, using mRNA as template to synthesize the first cDNA strand. So the significance of producing cDNA is to produce DNA molecule that contains only the coding region. It contains only exon. Okay, so cDNA can code for the synthesis of protein without the interruptions of intron, which is the non-coding region. So once you have produced the cDNA molecule, you can now insert the cDNA molecule in the plasmid. So cDNA is double-stranded DNA. You can now insert the cDNA into the plasmid, which is also double-stranded DNA molecule. You cannot insert mRNA into the plasmid because mRNA is single strand molecule. Okay, so once you have produced uh, recombinant plasmid that contains the information from the cDNA, now you can insert the recombinant plasmid into the host cell, which is the bacterial cell. So the bacterial cell is now transformed, and when the bacterial cell replicates, it also clones the cDNA, and the bacterial cell can also express the cDNA to produce the protein coded by the cDNA molecule. So when for example, when you want to produce a cDNA molecule, you have to isolate the mRNA molecule from the cell that produces the protein that you desired. Okay, you have, for example, you want the cDNA, you want the mRNA that can produce the protein insulin. Okay, so as you know, the organ that produces insulin is pancreas. So you have to isolate mRNA from pancreatic cell. So once you have isolated the mRNA from pancreatic cell, you insert into the test tube the first enzyme, which is reverse transcriptase. So reverse transcriptase will use the mRNA as template to synthesize the first strand of the cDNA, and then another enzyme will degrade this mRNA template, and then you add the enzyme DNA polymerase to synthesize the second strand of the cDNA molecule. So now you produce the cDNA molecule that you can insert into the plasmid to produce clones of the cDNA. And so that is the significance of cDNA. So you can also store the information of the cDNA in a cDNA library. Gene library can be described as, the answer is C, which is bacteria with plasmids containing a collection of cloned DNA. So gene library is also known as DNA library. So although the term, it has the word library here, it does not mean that it consists of collection of books like the normal library that we have. So it is actually a collection of information of DNA, okay, that is inserted into plasmid within bacteria. So there are two types of gene library or DNA library, which is genomic library, and then the second one is cDNA library. For genomic library, it contains DNA fragments from the entire genome of an organism. Okay, so as for cDNA library, it contains DNA fragments that only has the coding regions, because cDNA is synthesized from mRNA. So for eukaryotic cells, because during after transcription, the mRNA will undergo RNA processing to produce mature mRNA. So this mature mRNA will contain only coding region that the mRNA, that's why the mRNA is used as the template to synthesize cDNA. So cDNA library will only contain DNA fragments that contains only the coding regions. So what are the significance or the purpose of this gene library? One is to serve as collection and storage of genetic information for an organism. Second, the information within this library can be used for DNA sequencing to determine the sequence of DNA for certain gene, for example. And then the third purpose of gene library is for gene cloning. So you can select genes that you are interested in and clones and do gene cloning. Why tag polymerase is necessary for polymerase chain reaction (PCR) to occur. So you need to understand what is actually PCR. PCR is actually a method, okay, or a technique used to amplify or make many copies of specific DNA segments in vitro, or replication of DNA in a test tube. So basically, the DNA samples that you isolate or extracted from a sample of cells, the amount is really small. Okay, you have a small quantity of the DNA sample, so that's why you need to amplify or make many copies of the specific DNA segment, so that you can make further study on the DNA segments or the gene that you are studying. So in order for you to amplify the DNA molecule through PCR, you will require the enzyme tag polymerase. So basically, tag polymerase, they have the same function as the normal DNA polymerase, except that for tag polymerase, they are the enzyme that is extracted or isolated from the bacteria Thermus aquaticus, which is bacteria that lives in hot spring. So therefore, when you isolate and use this tag polymerase enzyme, it is really heat stable. Okay, it can withstand temperature that is really high, so up to 95 degrees Celsius. So during PCR, PCR has three stages, which is the first stage is denaturation. So when you add the enzyme into the reaction, obviously during denaturation, which can reach up to the temperature of 95 degrees, so this will cause the enzyme to be denatured. Okay, so that is the first stage, which is denaturation. Denaturation is to is the stage to break the hydrogen bonds between the two DNA strands and to separate the DNA strands to become template. And then after denaturation is the second stage, which is annealing. Okay, annealing, where you attach a primer to the separated DNA template, and then after that is the next stage, which is elongation or extension. So this is where the function of a tech polymerase will take part. Okay, so during extension, the temperature can reach up to, which is the temperature during extension is 72 degrees. Okay, 72 degrees is really high for the normal DNA polymerase. So at this temperature, the normal DNA polymerase will denature. So that's why you will need to use the enzyme tech polymerase. So the enzyme tech polymerase will add free nucleotides or new nucleotides uh, complementary, okay, to the original template strands. So that's why you will need tech polymerase to withstand high temperature during extension step. So that is the answer will be C. Which of the following DNA sequence can be recognized as restriction site? So restriction site are basically site on either the foreign DNA or the plasmids where restriction enzyme will cut at specific sequence. So one of the feature of restriction site is that they are palindromic. So what is palindromic? So palindromics are sequences on the DNA that are identical when they are read in the opposite direction of the two DNA strands. Okay, they read the same. Okay, they are identical in the opposite direction. So if you look at the list in the possible answer here, which one has the palindromic sequence? So the answer will be B. Okay, so if you look at the sequence, the top strand here of the DNA molecule, a segment of the DNA molecule which is at this restriction site, so it reads GGCC. And the bottom strand, when you read in the opposite direction, okay, opposite direction, so it also reads GGCC. So that is how you recognize a restriction site. They are palindromic. They read the same when they are read in the opposite direction. If you look at the other answer, for example A, okay, so the top strand reads GGTT and the bottom strand AACC. Okay, AACC. They are read in the opposite direction. Okay, so they are not the same. They are not identical. So that A is not the answer. Same goes to C and D. Okay, so when you read the sequence, the top strand is not the same as the bottom strand. The top strand is not the same as the bottom strand. So that is how you recognize a restriction site, by recognizing that they are palindromic. And another feature of a restriction site is that the length is really short, which is the length of a restriction site usually consists of 4 to 8 nucleotides long. How many molecules of DNA can be obtained from one molecule of DNA after 4 cycles of PCR? Okay, so during the initial stage of PCR, here it says that initially there is one molecule of the DNA. So it means that initially the process of PCR used only 2 DNA strands. Okay, 2 DNA strands as template to synthesize uh, or to replicate the DNA molecule during PCR. So you use this formula, which is 2n. Okay, so n is the number of cycle. So in this case, the question asks for 4 cycles. So basically the formula is just simple. So 2 to the power of 4, so the answer would be 16. Okay, so let's say if the question asks for uh, how many DNA molecules are produced after 30 cycles. Okay, so just insert into your calculator, which is 2 to the power of 30. So you will get about 1 billion of DNA molecules produced at the end of 30 cycles of PCR. So that would be the answer. So you just remember this formula, 2 to the power of n is the number of cycles. So the answer would be B. Which of the statement is incorrect for gel electrophoresis? Basically, gel electrophoresis is a technique to separate DNA fragments according to their size or mass. So in order to do this, you need to set up the gel electrophoresis setup. Okay, so it is something like what I've drawn here. So you will require electrophoresis tank. Okay, so the tank is not large according to your sample size. Okay, so if your sample size is small, so you will require a smaller electrophoresis tank. If your sample is larger, okay, you have more samples, so you will require a larger electrophoresis tank. So in order to run your DNA sample, so you will need to prepare agarose gel. Okay, so the agarose gel you need to prepare that contains sample wells. Okay, so these are sample wells. So this is where you will load your DNA sample. Okay, so after the agarose gel has been prepared, place the agarose gel inside the electrophoresis tank, and then you have to pour buffer solution into the tank. So the function of the buffer solution is to carry electric current across the tank, okay, from cathode to anode. So you need to connect the tank to a power source, okay, electric source, and where you have the DNA sample, this is where you connect the cathode part, okay, the negative charge part, and then at the end of the tank here, on this side, you will connect the anode. Okay, so this is because when you run for gel electrophoresis for about usually 1 hour, the DNA sample will migrate from the negative region to the positive region. This is because DNA is negatively charged. So obviously it will attract to the positive charge at the end of this side. Okay, here, right? So let's look at the possible choices of answer. So the question wants the incorrect statement. So the first statement, gel made of agarose or polyacrylamide is correct. So you can use either of these gel, agarose or polyacrylamide, to make the gel to run your DNA sample. And then B, separation of DNA fragment is based on size. Yes. Okay, so DNA fragment will separate according to their size. Okay, the smaller molecule of DNA will travel the fastest, and then the larger molecule will travel the slowest. So when you run for jet electrophoresis, you will take one where you will load your DNA ladder. So DNA ladder is some kind um, to compare the size of your DNA. It is like a standard. Okay. So for example, here you have your DNA ladder that indicates. Okay. So if your DNA sample is somewhere at the top, so the length of the DNA will be about 10,000 mass pair. So if it's at the bottom here, so the length of the size of the DNA will be about 100 base pair. So this is to compare, to know what is the size of your DNA sample. Okay, so therefore, uh, statement B is correct. For statement C, larger fragments move faster, but smaller move slower. Okay, larger fragment move faster. So obviously, it is not correct. When you load your DNA sample into the agarose gel, so it will create resistance for DNA molecule to move to move faster along the gel. So if your DNA sample is larger, so it will move slower. Okay, it will move slower, whereas the smaller DNA molecule will move faster. Okay, so this would be larger molecule of DNA compared to this one, which is a smaller DNA size. So the answer would be C. Okay. So let's look at the another choice, which is D. Negatively charged DNA migrates towards the positive pole. So that is correct. In gel electrophoresis, DNA fragment will be migrated to positive pole because DNA is negatively charged due to all the phosphate groups in the backbone. So you need to understand the concept of DNA molecule. So DNA molecule, as you know, it consists of double-stranded DNA, and the monomers for the polynucleotides are nucleotide, obviously, that consists of phosphate, sugar, and base. Okay, phosphate, sugar, and base. And the polynucleotides has the backbone that consists of repetition of phosphate, sugar, phosphate, sugar, phosphate, sugar. The phosphate part of the backbone, they are actually negatively charged. Okay, they are negatively charged. That's why DNA it is called as actually deoxyribonucleic acid. Okay, due to the negative charge of the phosphate. So when you run for gel electrophoresis, when you insert your DNA sample into the well of the gel, okay, so when you turn on the electric power, so the DNA molecule will migrate from the negative pole of the tank, okay, towards the positive pole. Okay, so because the DNA molecule is negatively charged, it migrates to the region that has a positive charge. So that's why you need to connect on this region is the cathode, and on this region is the anode. Which of the following is or are application of DNA technology? So let's look at the choices. So the first one, which is DNA recombinant technology, is used for gene therapy, production of insulin, and also production of vaccines. So the answer would be D. Basically, for gene therapy, it involves the introduction of genes, and these genes contain information or codes for the synthesis of certain proteins. Okay, so these genes are inserted into a modified virus, okay, which does not cause any disease. So this modified virus that contains the gene will act as a vehicle or vector, okay, to carry this gene into the infected individual or individual with a genetic disorder. So, for example, a genetic disease which is hemophilia A. So, hemophilia A is a type of genetic bleeding disorder. So, this is where the person with this disorder will cause continuous bleeding. Okay, continuous bleeding in the muscles, joint, and also internal organ causes spontaneous bleeding, okay, or prolonged bleeding. So this is due to the inability or missing, okay, missing clotting factors. So to treat hemophilia A, which is a type of genetic disorder, the gene that code for the normal protein for blood clotting, okay, the normal protein for normal blood clotting. So this gene is inserted into a modified virus. So this virus will not cause any disease to the human. So modified virus will act as a vehicle or vector to carry the normal gene that can produce the normal protein for normal blood clotting. So this gene is called as F8. Okay, so it is capable of producing normal protein for for normal blood clotting. So when you introduce the gene into the virus, so next the modified virus you introduce into certain region or organs, affected region or organ. So in this case of hemophilia, you introduce the virus to the liver. So the virus will then insert the gene into the cell's liver, and now the body can capable of producing the normal protein for blood clotting.